Thanks, Janice. Good afternoon, everyone. So today I'm going to talk to you about animals behaving badly and the role of carbon dioxide or CO2. So this work was done in collaboration with a number of people who are listed below. So we live in a time of change. So not only are we putting a lot of carbon dioxide into the atmosphere, which is causing problems like warming there, but this carbon dioxide is also going into the oceans. So the oceans act like this giant sponge and absorb about 1/3 of all the carbon dioxide that we put out into the atmosphere. What happens in the oceans is that carbon dioxide then reacts with seawater and forms carbonic acid. So this is the acid that actually causes the process of acidification. So as Greta mentioned, it's not only these kinds of things that are happening like ocean acidification or warming that's a problem, but it's a rate of change of these things that is also problematic. So for example, we're changing ocean chemistry 100 times faster now than any time in the last 650,000 years, and probably for much longer than that. So we do know about ocean acidification quite well because the carbonate chemistry of seawater is very well understood. So we know that we're shifting from an alkaline ocean into sort of slightly less alkaline ocean. So over the last 250 years, for example, the pH of our oceans has dropped by 0.1. Now this might not sound like very much because pH is measured on a log scale, but actually it's an equivalent to a 30% increase in the number of hydrogen ions in seawater, which is the same as a 30% increase in acidity of seawater. By the end of the century, we're projected to reach a 150% increase in acidity compared to 250 years ago. So we might imagine that these changes are likely to cause problems for marine animals. So particularly marine animals, like marine invertebrates that have calcium carbonate shells and skeletons. Now these animals can include mollusks like snails and clams and urchins, and also crustaceans. So marine invertebrates are really important in ocean ecosystems. So for example, they can be keystone species, they can be really abundant, and they're important in biogeochemical cycles, and they can modify the habitat. Invertebrates are also important for biodiversity, and we can see in the graph here, this is just an example on a coral reef. Now here we can see that the number of non-coral invertebrates species outnumber those of fish and the coral invertebrates by an order of magnitude. So invertebrates are incredibly diverse. We can also see, and this is a picture shown yesterday as well, that invertebrates are really important in terms of the trophic levels in marine systems. So for example here, not only on these lower trophic levels, the invertebrates really sort of make up these important lower levels, but they're also really important in these high levels. So here we've got a squid as an example. So invertebrates are super important in many of the interactions that structure marine communities. What we do know is that ocean acidification affects many life history traits in invertebrates, for example, survival, growth, development, and shell formation and calcification. But actually what we didn't really know much about until a couple of years ago is whether there might be any effects of ocean acidification on invertebrate behaviour. So I wanted to test this and I wanted to find out, might invertebrate behaviour be altered by the kinds of levels of CO2 that we're likely to see in the oceans by the end of this century? And if so, what might be the mechanism for this? So we might think of snails as slow-moving animals, but this is not true for some of our coral reef snails. Actually, they can move quite quickly. So this group of snails can jump backwards rapidly away from a predator, and they can do this because they have a modified foot or and a perculum, which is like the shell trap door. And they can move very quickly away from a slower-moving predator, such as this venomous cone shell here. So this is some research conducted at Lizard Island. This is just the normal escape response. So we can see here the prey snail on the left is jumping backwards quite rapidly for a snail away from its much slower moving predator on the right. So I place these prey snails in control and elevated CO2 conditions at those kind of levels that we're expected to see at the end of the century. Now, when I placed them in a large sandy test arena, in the absence of a predator, most of these prey snails did not jump, so this would be expected, so there's the grey bars here. Now, when they were placed in the arena in the presence of a predator, most of those control snails jumped, so this is the black bar here, the behavioural response. However, those snails that had been treated with CO2 in the presence of a predator, most of them did not jump, so you can see that this response is diminished here. So this was really a switching of this behaviour for the CO2 treated snails, and this resulted in about a 50% drop in the number of snails that did jump from the predator. So that, you might think of that as their decision making. But what about their strategy? So I also analysed the number of jumps that snails made and the distance moved. So when I considered all the snails, including those that didn't jump, I did see that there was a difference between control and then CO2 snails, and that was fewer jumps and a shorter distance moved. But actually, if I only analysed those snails that did decide to jump away, I found that there was no difference in the number of jumps between control and CO2 or the final distance away from the predator. So if they did decide to jump, they seemed to be moving just as far. But it did take them longer to think about it. So this is this thing called latency, and it's essentially the time taken to make their behavioural decision. So we can see here, for example, that the CO2-treated snails took nearly twice as long to decide to start to jump away from the predator compared to those control snails. Now, I also wanted to test the snail's physical ability. So to check that CO2 might have any response on their exercise ability, for example. So when we did this, we turned snails over, we measured the time that it took them to right themselves, to turn themselves the right way around, and also the number of foot flicks that required to do this using their special modified foot. So actually CO2 made no difference to this at all. So we also wanted to test metabolism in snails. Might CO2 change, for example, oxygen consumption? So we measured resting metabolic rate and also aerobic scope, because we could exercise these snails by getting them to jump. And actually we see that for the CO2 snails, there was no difference either in the resting metabolic rate or in the aerobic scope. So it really suggests that these CO2 conditions have no energetic cost for these snails, and that really physical ability is not being impaired by CO2, so something else is going on. So just to sum that bit up, fewer snails jumped in CO2, they took longer to think about it, and something else I didn't show you just was that they had this slightly altered trajectory of escape, so the path that they escaped along was slightly closer to the predator than those control snails. But importantly, their physical ability was retained. Now, there was a lot of individual variation observed in these snails, and so we might think about that leading to something like differential mortality in the future. So that's something that we'll be working on as an extension of this project. But what about other marine animals? So what about some of those higher trophic levels, for example? So what we wanted to test whether their complex behaviours could be altered. And so for this part of the research, we used cephalopods, and Blake Spady actually did this for his minor project here. And he's here today at the symposium, so if you have any questions about this part of the work, please ask him. So Blake measured the squid activity after exposure to sort of 3 different levels of CO2 control and then 2 elevated levels. And he also measured squid response to a startle stimulus. So the results from this show that actually CO2 made squid activity increase. So this is just a graph that shows the number of squid at rest, and white here decreases as CO2 increases on the x-axis. And those squid that exhibited active behaviours, so that were highly active, increased. And this can be measured by the number of line crosses, for example here, so we're showing as CO2 increases, then the number of line crosses increases as well. So for the defensive behaviour, actually CO2 seemed to increase the amount of inking that these squid did. So there was more individuals discharging ink in response to that startle stimulus. CO2 also increased the amount of jetting, so you can see here that the jetting response increased compared to that defensive posture. And also different kinds of body patterns were displayed. So for example, there was one body pattern here that was only displayed at the high CO2 level. So these changes could be either positive maybe or negative, and we're not yet sure how that will sort of play out in a species interaction role. So that's the subject of future research as well. So what about other groups? What about crustaceans, for example? And might closely related species be expected to respond in similar ways? So this was some more work at Lizard Island using some tropical crabs that inhabit a similar kind of niche. So for these two species, again, I exposed them to control elevated CO2 and actually measured their emergence time. So that can be thought of as sort of how bold they are, how quickly they emerge from their shell, using a standard disturbance. So for this first species on the left, actually CO2 treatment had no effect on their emergence time. However, for the species on the right, you can see that actually elevated CO2 made these species take much longer to emerge, so it kind of made them shyer. So we can see that, crustaceans seem to be altered, but actually similarly related species, so these are species within the same family, can respond differently. Now, because they live in a similar kind of niche, this could start to affect things like resource competition, for example. And actually differences between species is something, in response to CO2, is something that we see in fish behaviour as well. So given that these behavioural changes seem to be fairly widespread, what might the mechanism be? Well, in fish, for example, we think that it's likely to be the GABA-A receptor. Now, this receptor is a primary inhibitory neurotransmitter, and it's really widespread, so it occurs from hydra to humans, and GABA-A-like genes have been found in all animals really studied to date. So normally ions would flow this way into the receptor, but at elevated CO2 conditions, the theory is that they flow the other way. So this means that CO2, how CO2 could be responsible for altered behaviour. But what we can do is to treat animals with a drug called gabazine, which actually would stop binding of GABA to this receptor and then should restore normal behaviour if this is indeed the mechanism. So actually in fish, when we do this, we find that fish that have been treated with CO2, they actually spend a lot of time in their predator, in the scent of a predator, which is obviously a maladaptive behaviour. Actually, on their second run, when they have been treated with this drug, they spend a lot less time in that predator queue, so their behaviour responses go back to normal. So for fish, it does seem to be that this is the GABA-A receptor that's causing these behavioural changes. So I wanted to also test this in snails. So I had control and CO2 treated snails, and I had them in plain seawater and also in this drug treated seawater. Now, after they went into those seawater conditions, I placed the snails in a separate tank and I added some predator cue. Now, this predator cue should make the snails jump because it's the same as mimicking the presence of a predator. And we can see here that actually, indeed, that happens, at least for the control snails. So the control snails had lots of jumps in their tank when predator cue was added. However, those CO2 snails didn't jump as much, so their response again was impaired. However, when we add the drug gabazine and then we do this again and test it, what we should see is that the CO2 snails have restored behaviour, so they should jump just as much as the controls. And indeed, this is what we see. So when we have the CO2 snails that are also treated with this drug, they jump just the same as those control snails. So this suggests that in invertebrate snails, GABA-A like genes and GABA-A receptors might also be the mechanism by which behaviour is changing. So really, what does this mean sort of for our future oceans? Well, actually, these kind of sensory behavioural changes were previously rather unrecognised since a couple of years ago. So this sort of opens up a bit of a new research field for us to start to investigate the potential behavioural effects of ocean acidification on invertebrates. And given that many invertebrates have even benthic invertebrates that are later in life will have pelagic larval phases, for example, sensory cues for settlement, for example, could be really important. So for some groups, these changes could be important as effects of calcification, for example, choosing whether to avoid a predator or not could be as important as how well you can form your shell. So we can see that this also occurs across multiple taxa, like invertebrates as well as fishes, and might affect species interactions. So given that it might be this mechanism of GABA-A, we can see that these changes may be widespread in marine systems. So for future research, obviously we need to consider the effect of interacting stresses like temperature as well, and also consider the effects on the whole system, so the predators as well. And we need to try to better understand how this might affect, for example, also our seafood resources. Now, we also need to try to uncover the potential for acclimation and adaptation to help species overcome these problems. Although at least for fish it seems that acclimation might not be the answer, and then actually genetic adaptation is required. Thank you.